This video will demonstrate the CTD use case for connecting the dots in the Playbook Workflow Builder. CTD can connect the dots and aid in the interpretation of large and complex biological networks. With the generation of large multi-omic studies, the question arises of how we can interpret this data. CTD allows us to connect the dots within these networks and identify highly connected sets. These could be sets of genes or sets of proteins. We can then ask which of these sets are significantly connected in the specific biological context. Once we've identified sets that are highly connected, we can also ask which genes, for example, are guilty by association or used to connect these smaller sets of genes. These genes could also be used as novel candidate genes in further studies. CTD has been previously applied to identify biological signal in a variety of highly heterogeneous diseases. These include, for example, breast cancer, where CTD was used to identify differential markers of chemotherapy response in triple-negative breast cancer, and was able to identify small and informative biomarkers for the response of patients to different chemotherapy agents. CTD has also been used to diagnose rare metabolomic disorders using untargeted metabolomic profiling for specific patients. To empower the community to use CTD as a tool in their own datasets, we have deployed CTD on the Playbook Workflow Builder through the Common Fund data ecosystem. To utilize CTD on the Playbook, we first select the GeneSet input card. Here we can put in our genes of interest, including in this example, genes A, B, C, and D. We then have a choice of selecting either connecting the dots in the string network or connecting the dots in the Wiki Pathways network. In this case, string is a protein-protein interaction network, while Wiki Pathways is a community platform that's used to annotate biological pathways. Once we've selected one of these cards, in this case string, CTD will be performed with your gene set of interest in a weighted graph, in this case the string graph. The outputs of performing CTD will include the number of highly connected genes, the number of guilty by association genes or genes that act as connections between your genes of interest, as well as a graph that can be formatted in JSON form. We have also added a functionality where users can perform CTD on their own user-generated graph. For this functionality, we also start with the gene set input, but we then upload a gene count matrix. This gene count matrix will be used to create a custom adjacency file that is specific to your experimental conditions. This adjacency file will then be used in CTD along with your gene set of interest, and the response will once again be the number of highly connected nodes, the number of guilty by association nodes, and a graph which displays both of these. In summary, CTD is a method to connect the dots and identify sets of connected nodes in weighted graphs without computationally costly permutation testing. CTD can be used to identify subsets of genes of interest that are connected in string, wiki pathways, or user-created experimental graphs. And CTD is available for use through the Playbook and can be integrated into the Playbook's workflow along with their other tools. We would like to acknowledge the Playbook partnership team, the CFD community, and the NIH program officers. We would like to also specifically acknowledge the ERCC DCC, Preceda Bio, as well as the Links group. Thank you.